Okay.
So are we gonna have like quizzes on our homeworks like we did the chapters? Or
Nope. Now that we're onto the paper, it's totally different.
Okay.
Did you have a question? Okay. All right. Oh, you can't see. I can't do that. I have to put this on up there. Hold on. Stupid. All right, so what is the history behind this paper? This is not the first Hallmarks paper. The first one, I think, came out in 2001 by Robert Weinberg, who is kind of the current modern father of cancer. And then the person he worked with, that person came out with the next generation, a decade later, of the Hallmarks of Cancer. And that was 2011 that this paper was. So we're at 2023, and a lot of science has been done since then. So there is a follow-up to this paper, and if we have enough time, we'll get into it. But if we don't, this serves as a great basis for understanding all those hallmarks. Have you done the hallmarks already? Yes, you've done them all because they were in that book, but it probably wasn't explicitly said, here are all the hallmarks and here's the science behind said hallmarks. So that's why we always dive into this paper as, since this is an upper level course. Okay, so let me get this up and going. UC. Is that on? Okay. Okay, so you can see now, that's up. Let me see if this actually writes. Yep. Okay, so these are the 10 hallmarks of cancer. When you do your presentation, that's your final, you'll talk on each one of these. You can find versions of this picture all over the web. So when you pull pictures, just make sure you cite them at the end, or you can cite them on the slide. Included in this picture are different things that scientists have come out with to stop that hallmark. So when you see enabling replicative immortality, which is right here, this is the hallmark, the enabling implicative immortality, and this is the treatment, telomerase inhibitors. If we can pause the extending of our chromosomes, then slowly get shorter and we go into apoptosis and that cell dies, which is great if it's cancer. Okay, so that's the kind of the overall picture for all these hallmarks. Everyone uses a picture like this when discussing the hallmarks. Okay. So, sustaining proliferative signaling. No, wait, Sam, I'll start with you. What does signaling mean to you? Just to you. Ligand to receptor. That is a great definition of what we're looking at. So we're looking for a signal, and the signal is a ligand. Hannah, give me an example of a ligand that you know. Remember, passing is always available. It's first thing in the morning, we're just getting back into learning. You can pass. Brooke's ready. She's typing furiously into her computer trying to figure out the answer.
Brooke's not ready. Caleb, you got this one.
Yeah, we're going all the way to Caleb. Give me a ligand. Give me an example of any ligand that you know. Water is not a ligand. We don't have water receptors.
Hey, you took a guess though.
Yeah, exactly. Yep. All right, so Julie, how did we lose all three of them?
I don't know where Jillian went, but Paige went to the bathroom. So I'm assuming Jillian also went to the bathroom.
They're all doing something.
Maybe they're talking in the bathroom.
Yeah.
Should I go? Go something else.
But I hope Tyler's in a different bathroom. They're all talking in a bathroom. All right, Emma, go for it. Give me a ligand. Any ligand you know.
Like hormones?
There you go. Hormones.
Someone in here knows their stuff.
Okay. Christina, give me a hormone that you know that induces proliferative signaling. Estrogen does, yes. Estrogen is considered anabolic, meaning it's going to use a lot of things so that we can grow, so it would cause proliferative signaling. That is a good one. Lakin, can you give me another one?
Is EGF a hormone?
Yes, so epidermal growth factor, EGF, and sometimes it's called EGF-1. Epidermal growth factor, that's the ligand that we've learned the most about in this class. So essentially what cancer cells want to do is they want to send out a signal that will, or they want to receive a signal that will entice them to keep growing and growing and growing. Okay, Sam, back over to you. Why would cells ever stop growing?
Well, if they run out of like DNA, I don't really know what the exact like process is, but like if they like the DNA is like snipped, I guess.
Okay, so I said lack of nutrients was actually, which was not what you were getting at. You were getting on DNA getting smaller, and that would be chromosome shortening, which is why we have telomerase. But and that's exactly how a cell would signal itself to die. As it multiplies, its chromosomes get shorter and shorter. A short chromosome is a signal to go into apoptosis. So shortening of chromosomes. Hannah, what's another signal cells know, or how do cells know it's time to stop growing? And by growing, we mean size, but mostly we mean replication. Brooke's ready. But I want you to think about your cells, not your cells. Think about the HeLa cells that you're growing. Why will they, if they were normal cells and not cancer cells, why would they stop growing in the dish? Contact inhibition. All right, so cells that are normal like to spread out, talk to their neighbor, maybe touch their neighbor, but they don't necessarily want their neighbor sitting right on top of them. So contact inhibition is what will tell a normal cell to stop replicating. A cancerous cell will say, forget contact inhibition, let's all clump on together on top of this cell. Okay, so those are some key things for what does it mean to be proliferative signaling. So that's epidermal growth factor receptor, and then we have, why would they stop? Lack of nutrients, it'll tell it to stop. Shortening of chromosomes or contact inhibition. All right, good. Why is this not moving on? Okay, next one, draw the cell cycle. Just on your page, and then we'll discuss what you drew on your cell cycle, especially while I try to figure this out. Okay, most people will start with a circle, right? And then we say that is not a cell. That's the timeline of a cell, and it's circular, which it's not because cells die. But up top, we have mitosis. And then here to here, this is G1, this is S phase. And then G2. Okay, I don't know where I left off. Maybe, oh, Brooke, yep. G1. What happens in G1?
Growth.
Yeah, not wrong. Growth. Okay, Caleb, two things can happen in G1. Whoops. We got growth, but something else might happen in G1. It'll get a signal to do this.
To arrest?
It could, but that is not, under normal circumstances, it shouldn't arrest. So what else might happen in G1 other than growth? Starts with a D.
DNA.
Yeah, that's S, you're right. All right. Got any other things, or you want me to go? Emma. What else will happen in G1? Caleb has it now. What is it?
Well, I don't.
You sure?
I won't say it.
Emma, you want to pass?
Yeah.
Paige.
Duplicate organelles.
Nope, that's in G2. Good guess, though. You're not, that is a thing that happens. All right, Jillian, what else can happen in G1?
Starts with a D.
Yep. Pass it on to Christina.
I mean, I know it's parent for DNA replication, but it's.
But that's S,
Yeah.
right? Okay. Tyler? No. Lakin? Will we be back to see him?
Differentiation.
Boom! Differentiation. So, when we look at the cell cycle, most of the time we're talking about stem cells. So, it's the stem cells that need to continually replicate so that we get more stem cells so that we can grow more skin or make more red blood cells or make more white blood cells. When we talk about blood cells, there's many types, red, white, and the leukocytes. So you got neutrophils, eosinophils, basophils, all the other phils. There has to be a signal to cause them to differentiate from their stem cells. So in G1, they'll get signals to grow. They'll get signals to differentiate into their permanent form. Or stem cell will be told to start getting ready for replication. Okay, so now if we're past this G1 and we're starting to head into S phase, these would be the stem cells. And we need to ensure that we replicate them well so there's no DNA mutations. And then we go into G2, and I believe, Brooke, did you say, who said organelles?
Paige.
Paige, good job. Oh, now the two of you are the same to me.
Oh, that's everyone.
Replicate organelles. But the big one, oh, I think I spelled organelles wrong. The big one to understand is the difference between G1 causing the differentiation and then what will cause you to go into S phase for a stem cell. Okay, but in general, you have all of that down. You've learned it many, many times over. All right, so now we've got a new word, mitogenic. Hannah, we're back to you. What does mitogenic mean?
Like to continue growing and dividing, preparing for mitosis.
It's going to increase the levels of mitosis. That's all. A mitogenic signaling is telling the cell to go through mitosis. And again, these are probably stem cells that are going through mitosis. And okay, so we've got that down. Now we're getting into the stuff that your book started to explain, but we'll look at it a little bit further. So how does a cell know to move through the cell cycle? Well, all the things that we said before. There should be a signal. So in this picture, we're seeing epidermal growth factor as the signal. There needs to be a ligand, or sorry, that is the signal. There needs to be a receptor. And so in this picture, it's showing you the epidermal growth factor receptor. It's also saying that it's got another name, and it's called HER. HER1, there's HER2, there's NER1, NER2. But these are actually really important to understand what the receptors are, because in certain cancers, they're overactivated or they actually cancel them out. And so that's when somebody's looking at breast cancer and saying it's triple negative. It might be negative for the HER receptor. And so if that's true, it's harder to treat because if you don't have the receptors, you can't put a chemical to bind to said receptor. So it's harder to treat. All right, so in this figure, we have that epidermal growth factor, which is very typical. And why is it typical? Because it's one of these types of proteins. So what do we have here? Here's the cell membrane right here, so it'd be a giant cell somewhere below. The epidermal growth factor spans the membrane of the cell. So out here is where a ligand would bind. And so that's the side of the cell that's exposed to the other cells and the other extracellular fluid and things like that. And then down here, once you're past the cell membrane and inside the cell, then you have a tyrosine kinase domain, and basically that is going to phosphorylate other proteins. But that's the start of the signal. When you have a ligand bind to the outside of the receptor, that will cause the domains that are inside the cell to start phosphorylating other cells. So when we look at this is typical of a receptor that it has two right next to each other. So we have an epidermal growth factor receptor on one side and another one right next to it, and they dimerize. So here we have a dimer. And so there should be two epidermal growth hormones that bind to each side, causes the dimer, and that activates the kinase, and now it starts phosphorylating things that will help the cell go through the cell cycle. So if we go to here, this is showing you all the signaling that might be activated when you use epidermal growth factor. So we can see our favorite RAS, RAF. We also, the rest of them we haven't really talked about a lot. But in the end, you'll see that those signals activate a bunch of other proteins like RAS, RAF, which in this case will activate MEK and ERK. And in the end, you get cell survival. So this is showing you how a cell can receive a signal and how that signal starts activating other proteins to eventually lead to self-survival, growth, metabolism, replication, things like that. Anything that's going to cause mitosis. Okay, so how does a cancer cell make this signaling happen all the time? A regular cell is very, very, very controlled. A cancer cell just wants to have mitogenic signaling all the time. One way it can do it, which is probably the best way, the fastest way, autocrine, which means it sends out epidermal growth factor outside itself and it binds to itself. So essentially we've got a cancer cell, and it releases epidermal growth factor to bind to receptors. This is epidermal growth factor receptor. It will bind to its own receptor and cause it to go through mitosis. So that's an autocrine. That's the best way to keep it going. All right, where did I leave off? Hannah, did you answer?
Yes.
Okay, Oh, Brooke answered. So we're to Caleb.
No.
Oh, it's you.
Mhm.
Okay. What does cellular reciprocity mean? What do you think it means?
Like the ability to like interact or like signal with other cells.
Yeah, so what's going to happen in this case is that a cancer cell will tell a nearby cell, here's some growth hormone. Don't you want to send growth hormone back to me since I just gave you some? So they just talk to each other and tell each other both to grow. Okay, so that's how another way a cancer cell can continue to grow. The third way is by level of receptors. So again, here is the cell membrane, and we've got some receptors. Remember, they're usually in dimers. Okay, so there you can see 3 receptors, let's say, the tyrosine kinase receptors. If I've got that much epidermal growth factor, so those little dots are epidermal growth factor. So there's 8 ligands floating around, and I've got 3 receptors in the membrane. They will all get filled, and they will all have a mitogenic response. However, if I have another cancer cell, and it's decided to upregulate its receptors, now you can see it has a lot more receptors, which allows a lot more growth factor to bind, which will tell it to move through mitosis faster. So one way is to increase the levels of receptor in the cancer cell to become hyperresponsive to any bit of floating ligand around. Okay, the next way, also, while I said this was probably the best way, this is, and this are. These are even easier to do and better to do. So receptor becomes ligand independent. What that means is, let's go back to this image. See if I can erase this. Okay, what it's saying is that this setup where it dimerizes and activates doesn't need to happen. We don't need to have a ligand in there. It will dimerize and activate on its own. So auto activation without ligand. So essentially, it could always be on. Cancer cell just says, forget it. I don't even want to wait around for hormones to tell this cell to go through mitosis. I'll just activate by this mutation, and it'll always be on to go through the cell cycle. So there's that. Or the last one tells you just to have an active downstream signal. So this is considered upstream. It's the start of the signaling pathway. This is considered downstream. Okay, so the first steps of a signal are considered upstream. The final effect is the downstream, and the actions to get that final effect are the downstream. So if you could just have a RAS that is automatically on all the time, then I don't even have to worry about this top part. I don't even have to worry about the receptor dimerizing and autophosphorylation at all because we've activated RAS. RAS and RAF are common ones. SERC is another common one to always be turned on. Okay, so now we know several different ways that a cancer can cause mitosis. Either, we've got all these different ways. So it sends out its own hormone to its own receptor. It asks nearby cells to send it hormones. It increases that hormone receptor, making it hyperresponsive. It no longer needs the hormone. It just is automatically active at the receptor level, or even downstream from that, proteins have just become automatically activated. So constitutive activation means proteins that are always there are now always on. So constitutive just means what do you already have? You're not inducing, you're not making more. This is just, I already have RAS, and now it's on all the time. Seeing if I'm missing anything in my notes. Nope. Okay, so what can a cancer cell do to keep mitogenic signaling on? So before it was starting that signaling. Now we're talking about how do we keep it going on and on and on and on. And one thing would be inhibit negative feedback. So, trying to think about a good negative feedback loop. In our body, we have the hypothalamus, pituitary, and let's just say the adrenal. Okay, the adrenal gland is going to release cortisol, and that's the final product is cortisol. But to start that whole pathway, the hypothalamus had to release CRH. You don't need to memorize this. I'm just showing you a negative feedback loop. CRH to ACTH, which will lead to cortisol. And then cortisol will go back up to CRH and tell it to down-regulate, basically to stop. So that's an idea of a negative feedback. Once I've made the signal, I needed the cortisol. My body knew I needed the cortisol. But then there's a way to shut down the system when the body has enough cortisol. So enough cortisol has been released, and there's a signal up to the hypothalamus that when it reaches a peak level of cortisol, it shuts down that pathway. Thereby, in a couple minutes, that's actually a couple hours, cortisol levels will go down. So if we don't have that, if we don't have negative feedback in a cancer cell, and it's always being told to replicate, and there's no shutting the system down, then that's another way to keep it going. So there's always negative feedback in cells to stop them from having a over response, and here cancer cells figured out a way to bypass it. Okay, so let's look at the image here. Okay, we are looking up top at this tyrosine kinase receptor, okay? And it's binding some type of ligand, and here you can see the domains that have become active. Okay, so that part we already know. We know how to do that. What we're seeing are the pathways that will lead to mitosis and all that activity. What we want to do is mTOR is a great negative regulator that it knows to stop growing if there aren't nutrients around. Okay, so mTOR tells you there's no food here, so you really need to stop growing. And so you can see that it can go up until AKT. Again, it's another protein that we haven't really learned about, but it will go up and tell all of these pathways to stop. Okay, so mTOR, if mTOR has a mutation, if mTOR is a negative regulator and it has a mutation, it can no longer tell these signals to stop. That's another way a cancer will keep going, by telling the negative regulator, so the one that inhibits it, if we cut that one down and inhibit that, then it'll continue to go through the cell cycle, metabolism, and cell survival. Okay, so mutations in mTOR are really big in studying cancer because they are the negative regulator. Seeing if I'm missing anything else. Nope. Okay, now on your pages right there, right on that one, write down what you think your definition of cellular senescence is. Okay, so let's check. Let's check to see if you're right. Cellular senescence is basically stop of cell division. So you could also say cell cycle arrest. Okay. So now Caleb, I think we're at Caleb or we were at Brooke. Brooke, Caleb, Caleb, Caleb. Why would a cell go into senescence?
I guess like, could it be like contact inhibition of those needs to stop? Because I feel like since senescence more, sometimes it's when the cell gets old too.
Yeah, that's a correct answer. So we got age, we got contact. Anyone think of another one? Go for it, Paige.
Didn't it say, like, if it was receiving too many signals?
Over signaling. Maybe. Yeah, so like if you have hyperactivation of the MYC genes, that can cause it.
I'm in the exam, you asked a question like this, and I think there was one that was in the book, like a cancer cell will go into senescence and just like, it just won't die.
Yeah,
Like even if it stops dividing, it just doesn't die.
correct. Yeah, you can keep it in this like frozen state.
Okay.
Isn't that what happens in like, what is that, like dormant stages of like certain diseases, like when chickenpox turns into shingles and stuff like that?
I think that's when the virus gets encapsulated
Yeah.
enough where it is no longer in a cell that's dividing, so it can't replicate itself
Yeah.
until a signal. Yeah, I believe so. You were going to say something.
If it's damaged and needs repair.
Yep, damage. Damage is another great one. It has to decide, am I damaged or am I damaged too much that I should die? Or am I damaged just a little? Let's stop everything. Let's repair it, and then we'll decide to go through it. Yep. Anyone got another one? We've mentioned it already. There's a reason, like, so it looks at its external environment and what can it tell? It can't grow because something's missing. What might be missing?
Like nutrients?
Nutrients. So lots of reasons will tell a cell to go into senescence. Some cancer cells like that, there's a nice interplay of causing it to go into senescence, holding it there, and then deciding, should I continue to divide or is there too much damage or things like that. And we'll get to an instance like that. Okay. So next thing, because we'll come back to senescence in a second, but evading growth suppressors. So these are suppressors. Emma, give me any tumor suppressor you know. You can pass. No, never feel like can't pass. Paige,
I don't know why I can't think of
you got blankness. Jillian, I have no idea what you said. Yep. RB, retinoblastoma. That's one. Now the paper gave you another one, Christina. You know what it is?
Is it P53?
Yep, P53. All right, so those are the earliest known tumor suppressors. There's many more. We'll talk about one more. But RB and P53 are the most known. Sometimes you'll see it as TP53. The P stands for protein. T could stand for tumor or transformation, something dealing with the tumor. Okay, so if RB is on, you will not go through cellular division. If P3 is doing its job, you will not go through cellular division until it gives you the all clear. If a tumor can somehow render RB and P53 like mutations or silence them through phosphorylation or removing a phosphate, then the cancer can keep growing and growing and growing. So the second best thing it can do is evade growth receptors. You do not need to memorize this image, but it does show you the different types of things we're going to see. So this is the activation of P53. I know you can't see this, but you're just showing you how many different things stem off P53. So we've got some negative feedback. Remember how we talked about that mTOR? So you see here, mTOR stops the cell cycle inhibits the cell cycle production. We've got You got to repair. We've got angiogenesis, stopping that. So all of these things, apoptosis, that'll stop it. That'll kill it. And then other things that'll cause senescence. So P53 is huge in understanding cancer progression. It was once the first target for it, but now we target more things on the outside of the cells. Again, you don't need to memorize that chart, but you should know that P53 is a tumor suppressor and the things that it can do to stop a tumor. Inhibit all these different things will stop it. Okay, this is just the activation of RB. You've seen it before. So this is cyclin D with cyclin-dependent kinase. Those come together and will phosphorylate RB. And now it relieves its pressure, its negative inhibition of E2F. It's just a gene that causes S phase. So you already know this. You don't have to worry about it. In fact, don't even look at that one. Okay, so P53, we got all its functions. And then RB, we got the fact that it can be relieved of its inhibition by phosphorylating it with some of those cyclin-dependent kinases with cyclin. And then we got that contact inhibition that we've talked about. But who is it that talks about contact inhibition? And it is a protein called Merlin. Okay, so Merlin is a protein. I don't know why they gave it the name Merlin, but it causes all these different pathways. But what it's really doing is it has proteins that will notify the cell that it's hooked in to other proteins on another cell. So all of these proteins are sensing this interaction between the two cells. And once it recognizes there's interaction, it's going to tell a cell to stop. E-cadherin, that's just the protein that holds it all together. Again, Merlin is trying to check these connections. And if it sees it, then it's going to stop cellular growth. Now, if we can do something about Merlin and get rid of it, if a cancer cell can get rid of Merlin or cause some dysregulation, that's another way it would evade its suppressors. So again, we get different ways, and again, we're seeing mTOR again, that protein is important. We got another level of contact inhibition, LKB1. That's another protein. And again, what it wants to recognize is if there's contact going on, and if it recognizes contact going on, then it's going to stop cell growth and metabolism. If it recognizes contact is going on, it will induce autophagy, which is, well, we'll get to what autophagy is in a second, but essentially you're telling it to stop growing and start kind of eating in order to survive. Okay, so what cancer cells do is they don't want to stop. They want to keep growing. They don't want to have autophagy, so they do a loss of function of LKB1. So they essentially stop LKB1 from signaling, and therefore a cancer cell now will continue to grow and will stop any type of autophagic response. So contact inhibition, if we can stop that signaling, then you can increase the cancer cell to continue growing. Okay, next thing. TGF-beta. Normally you see it written like that, TGF-beta. Again, a huge protein to study in cancer. So TGF-beta, again, it's a ligand. So we're seeing it up here. And you can see it activates receptors, and in the end, what it does is it has a lot of anti-proliferative effects. Okay, so we're going to try to stop growing. All of that is in purple. Okay, so the good signaling is in purple, and it's going to stop proliferation if TGF-beta is working fine. Now, what a cancer cell wants to do, it doesn't want TGF-beta to work fine because it wants to proliferate. So it'll corrupt the signal at different places. But now, where we corrupt the signal, see how we're getting some of these green things to show up? All these green things will promote proliferation. So it has a shutdown, which is its normal pathway when TGF-beta works, and stops proliferation. If the cancer cell can corrupt its own TGF-beta signaling, they can actually turn it into a proliferative effect. Cancer cells are evil because they're changing what our cells normally do. They're taking normal signals and corrupting them. They're shutting down signals. They're hyperactivating certain signals. So cancer cells are very tricky on how they do their regulation, which means it's tricky to treat these cancers because you have to know what's on the outside of the cells in order to treat them. And there's so many different pathways. All right, on this page, write down the difference of apoptosis versus autophagy. They are different processes. What's the difference? Okay, apoptosis, everyone knows it as programmed cell death. Autophagy is cell eating organelles. Your own organelles. So this is a cell eating its own organelles. Okay, so with apoptosis, completely normal. What's, we're gonna go, I don't know, where was it? Jillian answered? Yeah, Jillian maybe said something. I don't know, Christina, did you say something? Let's say Christina said something. Tyler, programmed cell death, why would it happen?
I guess there's like a multitude of reasons that there's like the cell isn't ready to replicate. There's some sort of damage along the pathway that
So we can have
the cell is saying it's not good enough to be passed along.
damage can induce apoptosis if it's too far gone. Lakin, you got another answer?
Yes, but my other answer is better fit for autophagy.
Okay, all right, Sam, we're gonna come back to Lakin. Sam, you've actually, I think, already answered this. The reason why apoptosis would happen, I feel like you answered this.
Well, if there's, the DNA is like shortened.
Yep, chromosome shortening. Remember, as chromosomes replicate, they get shorter and shorter each time, and at some point it is too short, and it will say, I've been alive for so many cycles of stem cell replication that it's possible that there might be mutations. If I've divided so many times, I probably have 1 or 2 mutations. My chromosomes are super short, so I should just die. Okay, so programmed cell death does a thing called blebbing. I feel like we posted a video on it, but I can't remember. But essentially, you're going to take a cell that looks like that and slowly break it down, and it's going to bleb out into all these little tiny pieces, and then a macrophage will come around and just gobble it up, going, okay, thanks for dying, I'm going to eat the rest of these remains. Versus autophagy. Lakin, what's the reason autophagy would happen?
They talk about lack of nutrients.
Yep, that's probably the main reason. Lack of nutrients. And so, this is what happens in our own body too, kinda, to an extent. You know that if you're not eating, you will start to digest your insides, like muscle and things like that. So, first you'll move through your fat stores and all that fun stuff, but then you'll go into actually eating your own muscle. A cell, when it first gets the signal that there's not enough nutrients, will start to eat its own organelles. So think like you're eating a ribosome or something. Remember, a ribosome is made of protein. It helps make proteins, but it's also a protein. And so you just digest that protein, freeing up a little bit of extra protein that your cell may need to live just another couple hours, hoping that new nutrients come by. So autophagy is helpful, but it can also be harmful depending on when a cancer cell decides to override it. So autophagy, well, we'll get to a picture on how autophagy happens and the problems with it. But autophagy, instead of it doing what we call blebbing in apoptosis, it just swells up and bursts, just pops open. So apoptosis breaks down into tiny little things that get eaten up. It's called blebbing. Autophagy just bursts. No, that's not right. On my slide, sorry, that I just described necrosis. Autophagy is still just the cell eating. I was looking at a different slide, my bad, which is the next slide. There you go. Necrosis. Necrosis is when it needs, there's too much damage or whatever, and now it needs to, it's going to explode. So this is usually due to exhaustion, which a lot of you probably feel, or breakdown of everything happening in the cell. Again, probably a lot of you feel. Okay, so necrosis, again, can be good or bad for a cancer cell depending on what happens from it. But we'll look at first apoptosis. So apoptosis has two signals, two pathways. We have an extrinsic pathway and we have an intrinsic pathway. The extrinsic pathway is through the death receptor, great name for a receptor, death receptor. And it will cause essentially some of the same processes that the intrinsic. The whole purpose is to create those caspases to all come together and cause the breaking down of the cell, so the apoptosis. Okay, so that can come from the outside, or there's an internal signal, which is this, and again, it's telling us right here what might cause that internal signal. DNA damage, oncogene activation, stress, hypoxia, any of that will tell it to turn on and say, let's go through programmed cell death. The main regulators of the intrinsic signal are BCL2 proteins. So in general, BCL2 proteins can be more anti-apoptosis. And then we've got, where are they? I thought I saw them in here. Oh, these BAX and BAC, these are pro-apoptosis. So if you're a cancer cell, you want to figure out how to stop the BAX and BAD signaling so that apoptosis does not occur. Okay, so how do we stop the cell from going through apoptosis? Just inactivate that downstream signaling of BAX and BAD. Okay, autophagy. This is the image of autophagy that you can see. This is the nucleus, and what's going to happen is it's going to eat some organelles. So you can see that it decided to ingest its mitochondria into a lysosome. It breaks it down into little pieces so that you're freeing up sugars, amino acids, fatty acids, nucleotides, hoping that the cell in a couple of hours will get what it needs to survive even longer. Otherwise, if it's too far gone, then it will end up dying. So this is good if you're a cancer cell. You don't want to necessarily eat your insides and because you might end up dying. what's tricky about autophagy is it is cytoprotective. Okay, so it is trying to protect the cell. In chemotherapy, you're trying to kill the cell. If your cancer cells know to induce autophagy to a certain level to be cytoprotective, then it is essentially guarded itself from chemotherapy. So that's not good for us if we have cancer. If the cancer cell has figured out how to play with autophagy to be cytoprotective, the chemotherapeutic drugs won't work on their cancer cells. And that's not great.
Does it do that with radiation too?
Because it's leveling, it's measuring stress levels, and just think, oh, I'm going into stress. I need to get all the little free nutrients I can to be to keep myself alive. All right, and then the last one, necrosis. So tumor necrosis factor, we're seeing in this page, tumor necrosis factor, TNF. There's TNF alpha, there's a couple different other TNFs. This down here, the blue things are your tumor cells in general, mostly. So this is your kind of your microtumor environment. We haven't really talked about that too much, but it's the tumor cells and all the helpful cells around it. So we've got lymphocytes in there, you have a myeloid cell. I can also see bloodstream going through there, so which means there's lots of nutrients being spread throughout. Okay, so at some point, we're trying to kill the cancer cell. That's what we want to do. And so the malignant cell is kind of surveying itself, and it sees that it has DNA damage and things like that. So it might decide, okay, I should go through necrosis and die. When it decides to do that, it releases some signals that will call in lymphocytes. Okay, so you're a tumor cell, you're thinking about dying, and you release some signals that call in lymphocytes. Lymphocytes will come in, that's what we're seeing. Those green cells come over to the tumor and go, okay, you're dying, I'm going to try to help you. So while we think that the tumor cell is about to die because of its release of tumor necrosis factor, the lymphocytes, while it wants to kill it, it also at the same time is going to cause angiogenesis. It's going to cause all these things. It's going to help metastasis. It's going to help angiogenesis. So all of that survival, those elements will come back into play. So we tried to kill it, but the lymphocytes' involvement helped the cancer cell to survive. So tumor cells play this interesting game with necrosis. It's a very fine line to decide, should I die or should I try to die but hope that my white blood cells can help my microtumor environment and let me continue to grow. So this is a way a tumor cell can evade tumor suppressors, but at this point it's all the way down at the necrotic level. Okay, I think that's it. Are there any questions on the different topics? So you got to remember that now we can, you should, when you start to make your presentation, I still have to put up like what requirements for the presentation, but each hallmark you need to be able to explain with examples on how this is a hallmark of cancer. How does a cancer cell try to escape these so that it can continue to grow? The whole thing is trying to figure out how is the cancer cell so darn clever that cancer still exists today. All right, no questions? All right, then I will see everyone next week. Remember, you still got podcasts, and then we're doing 4 through 6 of the hallmarks.